Assalamu alaikum friends. Today we will learn about the microscope. The topics that we learn are microscopy, magnification, resolving power, and light microscope. I hope that you will learn many things from this lecture, and you can explain. You will able to explain the definition of microscopy, magnification, resolution, also the description and use of light microscope. I will also explain the electron microscope in my next video. My today's lecture, I will conduct this lecture in English, not in Urdu. But if you want this lecture in Urdu, I will also explain all the things, each and every thing, each and every word, each and every phrase in Urdu. Okay, our first definition is microscopy. What is microscopy? The microscopy let us, the word microscopy let a name microscope in our mind. The instrument used to see small thing is microscope. As you know that if you want to magnify a thing, you want to see a thing in large size, small, very small thing in large size, very small thing in large size, you use microscope, and this use of microscope is called microscopy. Okay, next definition is magnification. Increase in apparent size of an object when seen under microscope is called magnification. When we see a small thing under a microscope, and we see its we see that its size become increase. We see increase in its size, and this is called its magnification. Okay, next definition is resolving power. Resolving power, or we can also call it resolution, is ability, is a trendency. It's ability of an optical instrument to show how show two objects separate. Resolution is defined as minimum distance that 2 points can be separated and as distinguished as two separated points. With the help of lens, resolution can be increased. When we use lens, we can also increase the resolution. Okay, optical instrument. The word optical instrument is led to the light instrument, which in which light is used. Optical means light, in which light is increased. The physics in the optical instrument is use of light. The resolution of a human eye, our eye resolution is 0.1 millimeter, use 0.1 mm. Okay, next topic is light microscope. This is the picture of light microscope. It's a modern light microscope in 21st century. Okay, in light microscope, I will let you the description of light microscope. Magnification, its magnification power, the magnification power of a microscope, light microscope. Resolution power of light microscope, structure of light microscope, and use of light microscope. Okay, first I will define you the description of light microscope. Okay, a light microscope is a biological laboratory instrument. It's a biological instrument or tool. We can also say it a biological tool that is used to that uses visible light to detect and magnify very small objects, in which we can also say that in light microscope, light is used to detect or magnify very small objects, and magnify very small object and enlarge them. The specimen is called that the object that is seen, that is small object that is seen larger in the microscope, it is called the specimen. The specimen is normally placed close to the microscope lenses. It is placed close to microscopic lenses, so we can see it in large in size. Okay. Next, the topic is magnification power. The magnification power of the light microscope is usually 1500x. You can say x. It's a variable quantity. Okay, the resolution power. As I have defined you the magnification, the definition of magnification and resolution, I think that I do not have to, don't have to explain you again and again. So the resolution can be 0.2 nanometers. Oh, sorry, 0. Sorry, I'm sorry. I have tell you wrong. 0.2 micrometer. The resolution of light microscope is usually as can be 0.2 micrometer. Structure of light microscope. Light microscope have many components, but in this picture are many few components. They're like eyepiece, nose piece, objective, body, light and specimen stage, and aperture, diaphragm and base. The main components of light microscope are eyepiece, lens tube, objective, revolver, stage, table, condenser, fine focus, line focus, luminous field diaphragm, and light source. Also, the main thing is source. The main thing is light source and the lens. Okay, the base. The first microscope was the that was discovered, that was invented, was have only have a lens tube and the base and a light source. Okay, now I will explain you the use of microscope, how it is used. Not in use. This is the main. This is the structural diagram. Many things are here. Many things are described. Okay, the use you can see. Okay, next the uses of now uses of light microscope. Light microscope are used to study living cells and for regular use when relatively low magnification of resolution is enough to study the membrane dynamics. Okay, to measure the concentration of calcium and uh, calcium changes and protein interactions, determine the location of specific protein, determine the shape of organ, cell, intellectual structure, examine the dynamics of protein, study protein interaction or protein conformation, examine the ion concentration. These are the uses of the light microscope. Okay, one thing that I have forget to tell you is that light microscope can only define the cell membrane, only cell, but not the cell organelles. It cannot define the cell organelles. To define cell organelle, we use electron microscope, and I will tell you electron microscope in my next lecture. So please subscribe. So please, I request you to subscribe my channel and to have more videos about my lectures about biology. Please subscribe my channel and like my videos, share with your friends, gain your knowledge and your friends knowledge, and please I request you to subscribe my channel. And if you want any lecture in Urdu, any topic about any grade, class 5, 6, 7, or 9, 8, any class you want, please tell me. Please tell me in comment section. Okay, thanks for watching my video, and have a good day ahead.